Does aggregation between platelet and myeloid cells associate with elevated coagulation markers months after COVID-19? SARS-CoV-2 is the pathogen responsible for COVID-19, the cause of the pandemic declared by the World Health Organization in March 2020, with many negative consequences in countries around the world. This disease primarily affects airways, but is capable of generating neurological, respiratory, and cardiovascular consequences. Many of the consequences of COVID-19 are associated with dysregulation of the immune system during the defense response against the pathogen. One of the possible long-term consequences of COVID-19 is post-acute COVID syndrome, PAX, or long COVID, which is marked by the persistence of symptoms such as fatigue, chest pain, and thrombotic complications, among various others. These symptoms can persist for weeks, months, or even years after infection, and the mechanisms responsible for the persistence of symptoms are not well elucidated. Keeping in mind the association of long COVID with cardiovascular and thrombotic complications, as well as other coagulation-related changes, we hypothesized that patients with long COVID have alterations in markers associated with coagulation in myeloid cells, specifically with neutrophils and monocytes. Furthermore, considering the importance of platelets in the coagulation system, we hypothesized that these cells would be interacting with myeloid cells in the context of long COVID-19 differently from the interaction happening in healthy individuals. Seeking to investigate these alterations, we will collect blood samples from patients diagnosed with long COVID-19. Next, we will process the blood samples by lysing the red blood cells with FACS lysing, followed by washing and centrifugation of sample. Using antibodies conjugated to fluorochromes, we will analyze the cells through flow cytometry, a technique that allows the assessment of cellular characteristics such as size, granularity, and fluorescence. Thus, we will be able to evaluate the level of expression of proteins associated with coagulation in monocytes and neutrophils, such as tissue factor, the initiator of the extrinsic pathway of the coagulation cascade, and CD11B, which recognizes fibrinogen. Furthermore, we also assess the occurrence of aggregation of myeloid cells with platelets targeting a platelet-exclusive protein called CD41. Additionally, we intend to evaluate the secretion of plasma cytokines through ELISA, aiming to identify elevated levels of cytokines capable of amplifying the inflammatory and coagulation response. Finally, we intend to isolate monocytes and neutrophils and subject the cells to procoagulant stimuli in order to compare the response between cells from long COVID-19 patients and healthy controls. Moreover, we will analyze the supernatant obtained from the stimuli in this experiment for cytokine analysis and quantification. The plates where the cells were seated in the experiment will be analyzed by microscopy, where we will assess the adhesion of monocytes and neutrophils through the plate area, and the supernatant obtained in this experiment will be subjected to ELISA for analysis of the cytokines secreted in response to stimuli. Here are the results we expect to find through these experiments.